All right, I decided to make this a separate video because there's a lot involved here, and note what it says at the top. You must listen to this in order to get the functions. So what's going to come on screen here is only the labels of the structures, and you need to listen to my explanation of their functions and note the functions for each one of these structures. Okay, so we start here with an E. coli. This drawing represents an E. coli bacteria, which, if you'll recall, is an endosymbiont that lives inside your digestive system and helps you digest your food. It's a chemoheterotrophic consumer. Chemoheterotrophic, a heterotroph eats others. It's other feeding. Literally translate, hetero means other and trophic means to feed. So heterotrophic, feed on others. And chemo describes where it gets its energy. So it gets its energy from chemicals that it eats, and it gets its carbon from chemicals that it eats. It's a chemoheterotrophic consumer. We're going to start on the inside and work our way out. And what we see here is not a nucleus, because remember, prokaryotic cells do not have a membrane-bound nucleus. This is referred to as the nucleoid region. It's just the region of the cell where you are most likely to find the one circular chromosome that is the genome of the prokaryotic cell. So it is made up of DNA, and there are not a bunch of proteins associated with that DNA like we see in eukaryotic cells. And so its function is basically the function of DNA. And you learned in a previous unit that DNA functions in heredity. But to break that down even more, if you'll recall, it stores information, genetic information. It transmits or passes on that genetic information for expression, for gene expression, and it replicates that genetic information to be passed on to new cells or new individuals through reproduction. So those are the functions of DNA, which is basically the function of the nucleoid. We also have this little ring here that is also a ring of DNA. It's called a plasmid. And not all prokaryotic cells have plasmids, and when they do, there's usually more than one. So there'd be probably thousands of plasmids in a prokaryote that has plasmids. But that's, again, just a little ring of DNA, and it functions, so its function is the same as DNA. However, it also is involved in another function known as conjugation. Conjugation is a way for one prokaryotic cell to pass genetic information to another prokaryotic cell. And I'll describe that more coming up, because the pili here, these extensions of the plasma membrane known as pili, are also involved in conjugation between prokaryotic cells. But let's move on with these other structures and functions. So we have the cytoplasm. The cytoplasm, and if you'll recall, that's one of the things that we find in all living cells. DNA is in all living cells, cytoplasm is in all living cells. And the cytoplasm is made up, if you'll recall, of the cytosol, which is the liquid portion of the cytoplasm, and that's where all chemical reactions are taking place within the cell, is in the cytoplasm, especially in a prokaryotic cell, because there are not membrane-bound compartments, membrane-bound organelles where other chemical reactions can take place. So they're basically all taking place in the cytoplasm. All metabolic reactions take place in the cytoplasm, in the cytosol of the cytoplasm. All these little dots that are basically the only organelle, almost the only organelle within the cytoplasm, are ribosomes. All living cells have ribosomes because all living cells have to make proteins, right? So ribosomes are the site of protein synthesis. Another way of saying that is ribosomes are the site of protein translation, something that you should be very, very familiar with now. Just outside of the cytoplasm and surrounding the entire cytoplasm and lining the first layer here that we see outside of the cytoplasm is the plasma membrane. You can also call it the cell membrane, but I prefer to call it the plasma membrane. It's a more technical term. And it helps you not confuse it with the cell wall, because we're also going to be labeling the cell wall. It's a lot easier to keep them separate if you say plasma membrane. The function of the plasma membrane is as a selective barrier. In 2 words, the function is as a selective barrier. It forms a barrier between what's inside the cell and outside the cell, and it controls or selects what can go through from outside to inside or inside to outside. So it functions as a selective barrier. I'm underlining here all those things that you'll find in all living cells. Again, all living cells have DNA, all living cells have cytoplasm, all living cells have ribosomes, and all living cells have a plasma membrane. Just to make sure that that's very clear. So those are like the basic structures of cells. You'll notice that there's this structure that's coming off of the plasma membrane, probably formed by the infolding of the plasma membrane or the ingrowth of the plasma membrane. And I put here in parentheses that it's ER-like. And you're going to learn all about the ER, the endoplasmic reticulum, coming up in another unit, actually, in our next unit. But the endoplasmic reticulum functions in protein and lipid synthesis and processing. Protein and lipid synthesis and processing. And it's kind of mysterious what the mesosome does here in prokaryotes, but it's thought that the mesosome is ER-like in its function. Next we have the pilus. One would be a pilus. Pili is plural. And the pilus or pili are for attachment. So it helps the prokaryotic cell attach and stay where it wants to stay. It also functions during conjugation because a bridge can be formed between 2 prokaryotic cells between their pili. So in other words, the pilus of one prokaryotic cell will fuse with the pilus of another prokaryotic cell, and genetic information will be passed between the cells. That's an example of what we call horizontal transfer of genetic information. Horizontal transfer, as opposed to vertical transfer, which is basically reproduction. And the genetic information that will be passed during conjugation would be a copy of the plasmid. So it's just the plasmid DNA that would be copied, that would be replicated and passed through the pili bridge from one prokaryotic cell to another during the process of conjugation. So both the plasmid and the pili function during conjugation. The next layer out that we see just outside the plasma membrane is the cell wall. And it's made of a combination of protein and carbohydrate. It's a compound called peptidoglycan. Peptido referring to like the peptide bond that we find between amino acids making up a polypeptide or a protein. And the glycan refers to a carbohydrate. So it's a protein-carbohydrate combination cell wall, and it functions in support and protection. So it helps the cell maintain its shape and supports the cell and protects the cell. Outside of that, we have a layer of kind of like gelatinous carbohydrate material known as the capsule. You can think of it as like sticky jelly. And it's not found in all prokaryotic cells. Not all prokaryotic cells have a capsule. For example, if you'll recall Griffith's transformation experiment, where there was the S strain of the bacteria, of the pneumonia-causing bacteria, and the R strain of pneumonia-causing bacteria, and the S strain was pathogenic and caused pneumonia, and the R strain was not. This is the difference. the S strain had a capsule and the R strain did not. And we see this in a lot of disease-causing bacteria, this capsule. And just like the pilus, or the pili, help the bacterial cell or prokaryotic cell stick, the capsule helps them stick also. So if we're talking about pneumonia, pneumonia is a disease of the lungs, and the capsule would help the S strain pneumonia-causing bacteria, the pathogenic bacteria, to stick to the tissues of the lung and stay there as they reproduce and make you sick. So that's the capsule. Then, if you'll notice, we've got this extension. Again, it's not an extension of the plasma membrane, but it's anchored in the plasma membrane, and it's this long whip-like structure known as a flagellum. That's singular, flagella, and some bacteria have multiple flagella, so flagella would be plural. But it functions in locomotion. That's the fancy term for moving from point A to point B, moving from one place to another. In the case of E. coli being in an aquatic environment, they would swim. They would use their flagellum to swim. We find flagella also in eukaryotic cells. For example, sperm cells have flagella, but their structure is different and they function differently. In prokaryotes, the flagellum spins like a propeller, whereas in eukaryotic flagella, they whip back and forth, more like a fish swimming motion. But that's the flagellum, and it functions again. You want to use that term locomotion because you need to know it. It's a vocabulary term. Down here, we're looking at a cyanobacteria. This drawing represents a cyanobacterium. And there's only one structure that we want to identify within the cyanobacterium, because cyanobacteria, as being prokaryotic cells, they have many of similar structures that we find in the E. coli that we just labeled here. But cyanobacteria have this special ability to photosynthesize. They're photoautotrophic producers, and they were the first photoautotrophic producers on the planet. Because bacteria were first on the planet, they eventually evolved the ability to photosynthesize, and that started adding oxygen to the atmosphere and created the system we all live within, basically, the oxygenated atmosphere anyway. So all of this membrane that fills almost the entire inside of the chloroplast is the membrane that functions in photosynthesis. It's known as thylakoid membrane. And when we study plant cells and we study the chloroplasts within plant cells, we are also going to identify the thylakoid membrane. That's the membrane that has chlorophyll. Chlorophyll being the pigment that absorbs the light energy that allows these organisms to carry out photosynthesis. And so that's basically where photosynthesis begins, is in the thylakoid membrane. And so we can consider this cyanobacteria to be very chloroplast-like. It's similar to a chloroplast, and chloroplasts are similar to cyanobacteria. And that all brings up the idea of endosymbiosis again. In other words, the chloroplasts that are in plant cells were once endosymbionts that lived inside another prokaryotic cell, and eventually evolved to become the chloroplasts that are now in both photosynthetic protists, like algae, and plant cells. So those are the structures that you need to concern yourself with when it comes to prokaryotic cell structure and function. And the idea here is that simple cells can perform all the basic functions of life. Even though these cells are simple, relatively simple compared to eukaryotic cells, they still are able to carry out all the functions of life. And one final point, there are scientists that are trying to build cells from scratch, and they're trying to figure out, well, just what is necessary to have a living cell. So they are working with the simplest cells that there are to try to use them as a model to build a cell from scratch and have it actually be alive, believe it or not.